very, 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 very impressive. So I figured I would start, since most of my research is kind of in endocrinology, and I apologize in advance if in the beginning it's a little bit too basic, especially going through the posters. I realized, wow, these are like way, way, way more advanced undergrads than I would love to have in my lab compared to some of my graduate students, actually. So I'm going to talk to you today about the factors. Most of the studies we carry out in the lab are looking at the factors that control hormone production. And most of our studies are interested in the factors that control cortisol production. So I'll try to alternate between these. So all of these hormones, aldosterone, cortisol, estradiol, and strategically not posted here, testosterone, is all of these are produced, these are steroid hormones, and they're produced from cholesterol in different localized sites in humans. And unlike other hormones like peptide hormones or catecholamines where they're stored in vesicles and ready for release, these hormones are produced and then secreted. So they're not really the ideal molecules that are able to allow you to adapt very, very rapidly to any external stimuli. So they're secreted as they're produced. Because they're members of the endocrine family of mediators, they travel through the bloodstream, typically bound to proteins because they're lipids, and they act in a wide variety of physiological processes, primarily by changing gene expression. So this is something that happens over time. The steroids here, estradiol, cortisol, aldosterone, of course, these are ligands for a large family of nuclear receptors that either, many of these, when bound to the hormone, bind to DNA, change gene transcription and then subsequent translation, and then a physiological process. Although some of these, like estradiol in particular, have been shown to act and bind to plasma membrane receptors and activate signaling processes and change physiological processes that way. So classically, as depicted here, steroid hormones, when bound to, say, albumin or other binding proteins, after they travel through the bloodstream, they can either act at the cell surface, and typically these responses temporally are a lot more rapid than the classical responses where the hormone, whether it be estradiol or cortisol, would bind to the cytoplasmic receptor. That receptor would enter the nucleus, bind to DNA, allow for activated gene transcription, translation, and then some kind of physiological process. And production of these steroid hormones is typically in response to a very well-timed and well-controlled feedback loop that is initiated in the hypothalamus, where peptide hormones, for example, adrenocorticotrophic hormone or luteinizing hormone, they will be secreted from the anterior pituitary in response to cues from the hypothalamus that would allow, once they're released, they enter their target tissue, whether that be the adrenal gland or the ovaries or the testis. These are ligands for G protein coupled receptors, so they bind to the extracellular surface of the cell and activate signaling cascades that then lead to increase, in the case of adrenocorticotrophic hormone, that can increase cortisol, luteinizing hormone, and FSH or follicle stimulating hormone, increase testosterone and estradiol. So these, particularly my lab is interested in cortisol release, and it plays an important role in the body's ability to adapt to stress. So as shown here, unlike the responses that are mediated by catecholamines, primarily released from the medulla region, the inner region of the adrenal gland, where you get the rapid fight or flight response, cortisol is released and acts to allow the body to more adjust in a more long-term fashion to changes, and that is mediated by the release of corticotrophin-releasing hormone, which signals the pituitary to release ACTH. ACTH acts in the adrenal, and you get increased cortisol production. And the adrenal gland is very distinct in that, in addition to having this region where you get the production of epinephrine and norepinephrine, in distinct region of the adrenal, you have distinct regions, the capacity to produce distinct steroid hormones. So in the response to angiotensin II or potassium, the outermost region of the adrenal cortex, the glomerulosa, and this release of angiotensin II and changes in potassium allow for the body to retain water and increase blood pressure, you get increased production of aldosterone. In the middle region, the zona fasciculata, it responds to ACTH, where you get a production in humans of cortisol and in rodents of corticosterone. And then in the innermost region, you get the production of adrenal androgens, such as dihydroepiandrosterone, the function of which is not known, although it's the most highly secreted molecule from the adrenal, particularly in response to ACTH. So this picture right here depicts the biosynthetic pathway of all of the enzymes that are involved in the conversion of cholesterol to cortisol or cholesterol to aldosterone. So one thing that you can appreciate is that it's a multi-step process, number one, and that there are several members of the cytochrome P450 superfamily of enzymes involved. So in the first step, you get the enzymatic conversion of cholesterol to pregnenolone by one member of the cytochrome P450 family, and that pregnenolone enters the ER where it can undergo several reactions, and the terminal step to produce cortisol occurs in mitochondria where another P450 acts to hydroxylate 11-deoxycortisol to form cortisol. However, the rate-limiting step actually in steroid hormone production is actually the delivery of free cholesterol to the inner mitochondrial membrane. And that happens in humans primarily by the action of LDL, and the LDL receptor, you get uptake of cholesterol esters, that those esters are the cholesterol, esterified cholesterol is deesterified, and then you get that transport of this cholesterol to the inner mitochondrial membrane. So I figured I have to put some kind of clinical, since a lot of you are pre-med, for shame. A lot of you are interested in human health, and I should be too. There are many factors that contribute to aberrant adrenal function and lead to changes in or pathophysiological conditions. One of the most widely characterized, or perhaps one of the most widely characterized, is congenital adrenal hyperplasia. And that can happen when you have a mutation primarily in one or more of the enzymes, primarily the P450s, that are responsible for the enzymatic conversions that I showed you on the previous slide. And that leads to a lot of lipid droplet accumulation, so you get a lot of cholesterol accumulating. And depending on the severity and the enzyme that is mutated, you can get virilization syndromes that lead to masculinization and hirsutism. So this is actually a female that you get the changes in the width of the shoulders and a severe virilization depending on the extent of the disease and the ability of the adrenal to primarily produce lots of adrenal androgens, which in females are converted to testosterone in the periphery. Another very widely characterized syndrome caused by mutation or the inability of the adrenal gland to secrete normal amounts of cortisol is Cushing syndrome. And Cushing syndrome has actually been studied a lot over the last 5-10 years with the increase of the metabolic syndrome, where you have a combination of defects that are associated with primarily a Western diet. So in Cushing syndrome, the body produces aberrant amounts of cortisol. This can be caused either by a tumor that's in the adrenal or at a distal site, but you get this excess truncal obesity, you get osteoporosis, hypertension, hyperglycemia, and the hirsutism that primarily is not from the cortisol, but from the DHEA in females that is converted to testosterone, so you get excess hair growth. So as I was talking to the people earlier today during lunch, cortisol is produced in a diurnal rhythm. So early in the morning, in a circadian rhythm, you get the hypothalamus early when most of us are sleeping. You get the release of corticotrophin-releasing hormone acting to activate ACTH production, and that cortisol, when it's released, it binds to the glucocorticoid receptor in target cells. And the glucocorticoid receptor then binds to DNA and allows for changes in multiple physiological processes. Perhaps one of the most widely studied, and actually one of the most widely studied for its pharmacological potential, is the potent ability of cortisol and synthetic glucocorticoids to potently suppress the immune system. So cortisol therapies, cortisone, is used a lot in treating a lot of inflammatory disease states, including rheumatoid arthritis, asthma, and many other conditions. Cortisol, when bound to the glucocorticoid receptor, also acts in the liver to allow for increased nutrient production, and in muscle and adipose tissue to allow for increased production of nutrients. So my lab spends a lot of time looking at this signaling pathway, where after the ACTH binds to its receptor, it's a G protein coupled receptor on adrenocortical cells, you get the activation of adenylyl cyclase, increased production of cyclic AMP, and the activation of the cyclic AMP-dependent protein kinase, or PKA. And then ultimately, the adrenal gland will secrete lots of cortisol and lots of DHEA. But classically, this process has been characterized into two temporally distinct pathways. So you get a rapid, what's called an acute response in response to ACTH signaling, and that occurs to allow for increased uptake of cholesterol in the form primarily of LDL in humans, and that cholesterol be deesterified and moved to the mitochondria. And then a chronic response, which allows for the increased transcription, so the increased mRNA expression of all of the genes, not only the P450s, but all of the genes that play some kind of supportive role in production of cortisol and secretion out of the adrenal. So one of the receptors that plays a role in this ACTH cyclic AMP response is a nuclear receptor called steroidogenic factor 1. And a lot of time is spent in my lab looking at mechanisms regulating transcriptional regulation and how signaling pathways lead to changes in gene expression. And SF1, as we call it, is a nuclear receptor, so it's structurally in the family of the estrogen receptor, the vitamin D receptor, thyroid hormone receptor. But unlike other nuclear receptors that, say, in the picture I showed you earlier, where they bind the ligand in the cytoplasm and then go to the nucleus, SF1 is primarily expressed in the nucleus, and it acts as it binds to DNA as a monomer. What we do know about SF1 is, if you want to find out the function of a protein, one of the great ways to figure that out is to get rid of it. So in targeted disruption of SF1 in the mouse, it's an embryonic lethal phenotype. And what was shown is that if you look at the wild-type female with the adrenal and the ovary, in the absence of SF1, you get the lack of the adrenal, so you get embryonic lethality because of the lack of aldosterone, and the oviduct that fails to differentiate into a functioning ovary. And in the males, in the wild type, you see the presence of the epididymis and testis, and in the knockout male, you don't see an adrenal, and the oviduct is not degraded, and so what you get is what's called a male to female sex reversal, because the oviduct is retained, the Wolffian duct never differentiates into the epididymis or testis. So SF1 is not only important for the production, the increased expression of the genes that are required for hormone production, but also for development of the endocrine system. And what we also know is that SF1 has been shown to be regulated by its ability to be phosphorylated, to be ubiquitinated, to also bind to ligands. And one of the projects that we had spent a lot of time carrying out over the last about 5 or so years is to try to identify ligands for SF1, because previously it was called an orphan receptor because endogenous ligands were not known. So what was known is that if you actually express the ligand binding domain, the part of the receptor that should bind to a ligand, in E. coli, and crystallize that structure, several labs, 3 labs independently actually crystallized SF1 and found that it bound to phospholipids. And that suggested not only that the actual pocket is able to accommodate some kind of molecule, but that the molecule actually was found to be required and important for the ability of the receptor to activate gene expression. So what we did in the lab is we purified, and I say we loosely, meaning one of my former graduate students did, is purify SF1 from lots and lots and lots of adrenal cells, and then actually carry out mass spectrometry to actually identify lipids that were bound to the receptor that was purified from either cells that were untreated or cells that were treated with an analog of cyclic AMP. And we use this analog to activate the ACTH signaling pathway. And what we found is that SF1 is bound to a molecule called sphingosine, a sphingolipid, in control cells, and the amount of sphingosine bound to the receptor when we stimulated and activated the cells was decreased by about half. And that suggested to us that perhaps this molecule was not acting to activate SF1, but perhaps it was repressing the function of the receptor. And we went on to show that this sphingosine is actually an antagonist for SF1, because if we look at one of the genes involved in steroid hormone biosynthesis, CYP17, which is one of the P450s, you can see that if we treat the adrenal cells with dibutyl cyclic AMP in the presence or absence of sphingosine, and then isolate the RNA and do real-time RT-PCR, you get an increase in the expression of CYP17 transcript that can be attenuated if we also treat the cells with sphingosine. And then if we actually, sphingosine can be made by the one family of enzymes called ceramidases. And if we silence this members of this family of enzymes, if we silence ASH1, independent of cyclic AMP, just reducing the levels of ASH1 allows for increased CYP17 mRNA expression, although not as highly induction of mRNA as you see with dibutyl cyclic AMP. So sphingolipids became a new important friend to us after seeing this. And we've spent a lot of time since then looking at the factors that control sphingolipid metabolism. So sphingolipids are a large family of molecules. Many, originally when they were first characterized, were thought just to play a role like cholesterol, just to be important structural components of the cells. But many of these molecules have very, very important biological bioactivities in a wide variety of cells. So they're produced from serine and palmitoyl-CoA when you, and one of the central molecules that's produced is a molecule called ceramide. Ceramide is a very potent apoptotic inducer that can cause cell death. However, when ceramide is converted to sphingosine, and that sphingosine phosphorylated to form sphingosine 1-phosphate, sphingosine 1-phosphate is a very potent mitogen that increases cell proliferation and also cell migration. And actually, many pharmaceutical companies, there are several drugs in clinical trial right now that are targeting the decreased production of sphingosine 1-phosphate as a way to kill various cancers. So our hypothesis was at the time, in trying to figure out how sphingosine was modulating the actual function of SF1, we knew that sphingosine bound to the receptor, but one thing we did know is that SF1 is a nuclear protein. So if it's going to, any lipid that's going to be made in the cell either has to be made in the nucleus where SF1 is because it didn't spend time in the cytoplasm, or it had to be transported to the nucleus. So our hypothesis was that these sphingolipids that I'm going to tell you today, or phospholipids that I'm not going to talk about, which actually activate the receptor, are transcriptional, the enzymes that make these are actually acting in the nucleus to control gene expression. So this was our model pathway where ACTH signaling would lead to activated cyclic AMP, and production of cyclic AMP, the activation of pKa, and the activation of a kinase called diacylglycerol kinase. And we found previously that this diacylglycerol kinase formed phosphatidic acid, which bound to SF1 and activated the receptor. And in converse, ceramidase, which actually led to the production of sphingosine, repressed the receptor. But we postulated in our model that all of these changes in the gene expression were mediated by the ability of the nucleus to locally produce phosphatidic acid and sphingosine. So our hypothesis was that these molecules were being produced in nuclei, and that the nucleus was actually some kind of hub for the synthesis of bioactive lipids that would act to control gene expression programs. So we first sought out to try to determine what enzymes in the sphingolipid biosynthetic pathway were actually expressed in nuclei. And we used the H295R as an adrenocortical cell line, mammalian cell line, and looked using both Western subcellular fractionation and Western blotting and immunofluorescence to determine which of these enzymes, the serine palmitoyltransferase, the synthases, the members of the synthase family, which of these enzymes were present in the nucleus. And what we found was, as shown by Western blotting and by staining, that most of the enzymes involved in de novo sphingolipid biosynthesis were not present in the nucleus, except for one subunit of the serine palmitoyltransferase, but the other subunit was not expressed. and that most of the enzymes involved in sphingomyelin production, a higher, more complex sphingolipid, were not also present in the nucleus of these cells. However, in contrast, when we looked at for the presence of enzymes involved in the conversion of ceramide to sphingosine, we saw that ASH1, the enzyme that if we knocked it out by RNA interference, if we knocked it down, we could increase CYP17 gene expression. That enzyme actually was present in the nucleus, as well as ASH2, another family member. SF1, as I mentioned before, is a nuclear protein. It's not expressed in the cytoplasm. And as controls, we also looked at the expression of a mitochondrial protein that's expressed in the adrenal called STAR, which is not in the nucleus, and CYP17, which is not in the nucleus. So we then, if ASH1 was present in the nucleus, and we knew that if we knocked it down, we increased gene expression, we wanted to try to figure out the mechanism that by which this enzyme actually acts to control hormone production. So what Natasha Lukia, a former graduate student in my lab, did was to engineer a cell line, a TET-inducible cell line, where she allowed for the made a stable cell line where all the cells expressed a plasmid that when the cells were treated with tetracycline, they would express a short hairpin RNA that would bind to ASH1 mRNA and decrease the actual translation. So when she treated with tetracycline, she would decrease specifically the expression of ASH1 in these cells. And this, on the left-hand side, is just the steroidogenesis biosynthetic pathway and the enzymes that we looked at that are both transcript levels and protein levels to see what the effect was of silencing this gene on the ability of these cells to produce cortisol. So if she looked at the wild type, if you treat with dibutyl cyclic AMP as our control, you can increase the mRNA expression of most of the genes involved in steroid hormone biosynthesis. So you get an increase in CYP17, CYP11A1, all of these genes that are important for the conversion of cholesterol to cortisol or cholesterol to DHEA. However, if she knocked down ASH1, what she saw was an increase actually in the basal expression, even in the absence of cyclic AMP stimulation, an increase in the basal expression of STAR, which is important for transporting cholesterol to mitochondria, the CYP21, and important in converting 11-hydroxyprogesterone to 11-deoxycortisol. So all of these genes, the mRNA expression was increased. So in order to determine if this increase in transcript actually translated to an increase in function of these, the capacity of the cells to secrete more cortisol or DHEA, she also looked at, by Western blotting, at the protein levels. So if she looked at compared the wild type cells to ASH1 knockdown cells, you see an increase in the expression of the CYP17 or P450C17, P450SCC, which is here. All of these genes were increased in expression when she knocked down the acid ceramidase. And that suggested that the acid ceramidase was playing an important role in repressing basal cortisol production. And in doing so by decreasing the ability of the nucleus to produce sphingosine. And the last thing that Natasha did is to actually quantify the amount of hormone that was being secreted from these cells. So using an ELISA assay, she actually collected the media from cells, wild-type cells or the ASH1 knockdown cells that were either untreated or treated with dibutyl cyclic AMP. And what you can see is if she quantified cortisol levels, in the wild-type cells, you get an increase with dibutyl cyclic AMP in the secretion of cortisol. You also get an increase in the secretion of the androgen DHEA or dihydroepiandrosterone in the wild type when you treat with dibutyl cyclic AMP. However, if she knocked down the ASH1, you can see that even in the absence of cyclic AMP stimulation in the black bars, when you compare wild type to knockdown, she saw a basal increase in the production of cortisol. So these cells basally secreted more cortisol than the wild type cells, and that was further increased when she stimulated the cells with dibutyl cyclic AMP, particularly for the DHEA, the androgen. So if you compare wild type to knockdown, you see in the presence of cyclic AMP a huge increase in the capacity of these cells that lack ASH1 to secrete more adrenal androgen. So since SF1, again, to remind you, is in the nucleus, the next thing that she wanted to do was try to figure out, well, how is ASH doing this? Does SF1 bind to ASH1 in actually mediating its ability to affect cortisol production? And to determine if the actual proteins interact, she did what's called a co-immunoprecipitation assay, where she transfected the adrenal cells with either a flag-tagged ASH1 and or a GFP-tagged SF1. So she also used an ASH2 as a control, because ASH2, she showed, if you can recall a few slides earlier, was also expressed in the nuclei of the adrenal cells. So if she transfected with ASH1 in the presence or absence of SF1, and then actually looked for purified the lysates and immunoprecipitated with an antibody that for GFP, which would detect the bound SF1, you can see that ASH1 only precipitated when in the presence of SF1, so that these proteins actually co-immunoprecipitated from the cell, but ASH2 did not co-immunoprecipitate when she tried to purify with the GFP-tagged SF1. So that suggested that the two proteins actually interact. And the next question that she wanted to ask was that there are multiple complex ways to regulate gene expression, and those involve many complex regulatory mechanisms that allow for a large variety of proteins to either activate or repress transcription. And they do so when shown here as depicted by a generic promoter that is bound by a nuclear receptor. Of course, if it were SF1, it would just be one nuclear receptor instead of a dimer. So this would be a classical model for, say, the estrogen receptor. When estradiol is bound, the nuclear receptor binds to DNA, and it's the presence and the coordinate action of a large variety of other proteins that bind to the nuclear receptor that allow for it to either activate or repress transcription. And proteins that are shown in green that activate transcription are called that help activate transcription are called coactivators, where proteins that are shown in red that help repress transcription are called co-repressors. And coactivators act by changing the chromatin structure and allowing for the nuclear receptors to more effectively communicate with the basal transcriptional machinery, so that histone acetyltransferases would acetylate lysines residues on histones and allow for a more decondensed state that facilitates communication between the typically distally bound nuclear receptor and the basal transcription machinery, the RNA polymerase II complex. Whereas repressors, such as the histone deacetylases, will remove these acetylation marks and facilitate a more condensed DNA structure that will prevent the nuclear receptors from even interacting with the DNA, and also prevent communication between the bound nuclear receptor and the basal transcription machinery. So Natasha's hypothesis was that the ASH1 was probably acting as a co-repressor, and is in doing so not just interacting with SF1, but interacting with SF1 when SF1 was bound to DNA. And one of the ways in which we have used to determine what proteins are bound to DNA is a technique called chromatin immunoprecipitation, or CHIP. So not CHIP like microarray chips, but CHIP like chromatin IP, where we actually incubated the control cells or cells that were stimulated with dibutyryl cyclic AMP with formaldehyde for a short time. And that formaldehyde acted to cross-link either DNA to proteins and proteins to proteins, and then we isolated these cells, sonicated them to fragment that chromatin, and then immunoprecipitated with an antibody to either SF1 or to ASH1 or to the RNA polymerase or to an acetylated histone or any of the targets that protein targets that we were interested in. After that immunoprecipitation, we washed away unspecific binding and actually digested the protein away and purified that DNA and carried out qPCR and agarose gel electrophoresis to determine what regions of the DNA were bound by SF1 and or ASH1. So what Natasha found was that ASH1, as we had shown before, or SF1, and the polymerase, the RNA polymerase 2, are bound to the CYP17 promoter only when the cells were treated, in an increased amount, I should say, when the cells were treated with dibutyl cyclic AMP. The ASH1 was also bound to the promoter and increased at an increased level when the cells were treated with cyclic AMP. We also found that the methylation, the trimethylation of histone 4, or lysine 4 on histone H3 was actually increased. And that's a mark for a gene that's actively being transcribed. So if you immunoprecipitate with an antibody that detects that specific trimethylated pattern on lysine 4 of histone 3, you can determine if you have a promoter that's competent to be activated or undergoing activated gene expression. So she is gone now, sadly, from my lab. She was very productive, published so many papers, I miss her. But she, basically, what we concluded from her studies is that these ASH signaling, it activates nuclear sphingolipid metabolism. And this to us was a novel concept because the nucleus was not thought to be an area where you have the metabolism and production of bioactive sphingolipids or phospholipids, which someone else is actually working on. We also found that ASH1 interacts with SF1, and importantly, that interaction occurs when it's on DNA, and that ASH1 plays an important role in repressing both the expression of the genes that are involved in hormone production, so that when we silence that ASH1, we get an increased gene expression and an increased capacity of the adrenal cells to secrete both cortisol and DHEA. So I wanted to tell you a different story because transcription is interesting, but in the afternoon I wanted to talk a little bit about some studies that we've been doing looking at factors that are involved in more rapid changes in the cell when they're treated with ACTH or agents that mimic cyclic AMP. So one of the things that we were interested in, and we weren't interested in this in retrospect, we were demoing a microscope. But that's not what I told the NIH when I wrote this grant, actually. But we were demoing a microscope, and we wanted to, it allowed real-time video imaging, and we wanted to look at something cool. So we treated cells with all kinds of different cells, breast cancer cells, liver cells, adrenal cells, just anything we had with all kinds of agents, and looked for any changes that we could see in live cells by video imaging. And we came up with something interesting, and hopefully we'll continue, I know we'll continue to study at least for the next several years. And that involves one rate-limiting and interesting phenomena that we never actually considered in the lab until demoing this microscope, and that is the first step in hormone biosynthesis, steroid hormone biosynthesis, occurs in mitochondria. The pregnenolone actually somehow enters the ER, so it can either become 17-hydroxypregnenolone or progesterone. And in order to get either cortisol or aldosterone, the active hormones, the substrates, the 11-deoxycortisol or deoxycorticosterone, have to go back to mitochondria. So that suggested some kind of need for communication between the mitochondria and the ER in allowing for this one pathway to occur. And actually, this type of discontinuous or requirement for two organelles to communicate actually happens in phospholipid biosynthesis also. So what we did, or we meaning a postdoc in the lab, Donghui Li, is she stained the adrenal cells with mitotracker red. So the adrenal cells the mitochondria in these cells were actually, can be visualized as these red markings. And then what she did is actually treated the cells with ACTH and carry out real-time video imaging. And what she noticed is that the mitochondria in this particular cell, and only in the adrenal cells when we stimulated, actually started moving around in a pattern that we never appreciated, never knew happened. And she did this not only with ACTH, but also with dibutyl cyclic AMP. As I mentioned before, we treated several other cell lines with other agents known to activate various physiological processes in those cells, and we didn't see the same actual phenomenon. And if she quantified this, we could see that the ACTH and dibutyl cyclic AMP actually stimulated an increase in the rate of mitochondrial movement. So this was all cool and nice, very interesting visually, But scientifically, one of the things we wanted to see is if it was important, how could we stop it? And what is known, there was a lot known that we didn't know about at the time about the role of the cytoskeleton in controlling the positioning of organelles in a cell. And mitochondrial movement actually is very well studied, particularly in neurons, where the mitochondria move along microtubule tracks to allow for ATP to be delivered to distal regions of the cell. And shown here is a microtubule, here in the yellow tubes, and a picture of a mitochondria tethered. So there was precedence for an important role of the cytoskeleton in allowing for positioning of organelles in a cell. So what we did next is to determine if we treated cells with agents that alter the cytoskeleton, so either polymerize, cause the depolymerization of those microtubules into tubulin, or stabilize the microtubules so that they can't be depolymerized, or cause the depolymerization of the actin filaments, or stabilize the actin filaments, what would be the effect on that mitochondrial movement? So as shown on the left panel, Taxol or paclitaxel actually disrupts actin filaments, and even in the absence of cyclic AMP stimulation, so there was no dibutyl cyclic AMP, no ACTH, we saw an increase in that mitochondrial movement when she just treated the cells with Taxol. But when she treated the cells with an agent colchicine that inhibited microtubule polymerization, you can see that these the mitochondria were just pretty much immobile. They did not move, whether she treated with dibutyl cyclic AMP, ACTH not shown here, or other stimuli that we know activate either cortisol or aldosterone production. So when she quantified all of that data, what she got is that if you treat cells as she's shown before with dibutyl cyclic AMP, you get an increase in that movement. And if you inhibit the polymerization of microtubules, either with colchicine, shown as COLC, or nicotazole, you prevent the ability of dibutyl cyclic AMP to increase that movement. And then if you inhibit the polymerization of actin filaments with Taxol or paclitaxel, or stabilize microtubules by using latrunculin B, you can see even in the absence of cyclic AMP stimulation, you get an increase in that movement. So to establish a functional significance for this just observation that we were seeing, she collected the media from cells that were treated with these polymerization inhibitors and stabilizers, and we quantified the levels of cortisol or DHEA that were secreted. So if you treat cells with already known with dibutyl cyclic AMP, both cortisol production and DHEA production is increased, and that also happens when you treat cells with ACTH. However, if you actually, in the cells that were treated with dibutyl cyclic AMP, if they're exposed to either colchicine or nicotazole, which inhibited microtubule movement, mitochondrial movement, I should say, and prevented the polymerization of the microtubules, you can see that they did not produce the cortisol in response to dibutyl cyclic AMP, as shown in the dotted lines, but they produced a lot more DHEA. In contrast, if you treated with the agents that the paclitaxel that increased or prevented actin polymerization, you see an increase in cortisol even in the absence of dibutyl cyclic AMP, where you saw an increase in mitochondrial movement even when the cells were not stimulated with dibutyl cyclic AMP. So since the pregnenolone has to enter the mitochondria, or leave the mitochondria and enter ER to become DHEA or any of these other metabolites, and the 11-deoxycortisol has to leave the ER and go back to mitochondria to become cortisol. And we saw no changes, no effect of the inhibitors or stabilizers on the concentrations not only of DHEA. In any event, we saw an increase in DHEA, but no decrease in DHEA, or not shown in progesterone or deoxycorticosterone or any of these agents, other metabolites that we measured by mass spec. The only thing that we saw that was impaired was an ability of the cells to produce cortisol when we inhibited microtubule polymerization. We've suspected, but perhaps that movement may play a role in allowing the movement of 11-deoxycortisol back to mitochondria so it can be converted to cortisol. And one of the signaling pathways that we investigated was the Rho signaling pathway, because it is well known to mediate the changes in the cytoskeleton in response to either activation of receptor tyrosine kinases like the EGF receptor or G protein coupled receptor signaling like the signaling that happens in response to ACTH. And Rho, these are GTPases, small GTPases that allow for, in the inactive form, are bound to GDP. When they're active, they are bound to, you get an exchange of that GDP for GTP. This activated Rho can then act to allow for a wide variety of processes, primarily related to changes in cell shape and migration, and that is mediated by its ability to interact with other downstream proteins. So what she found is that if she treated cells with ACTH, she got a rapid activation of Rho, meaning that the Rho in the cell was actually Rho that was GTP bound. And that when we, not shown here, if we transfected these cells with a dominant negative Rho, we also were able to inhibit cortisol production and the cells produced a lot of DHEA instead. Rho actually mediates its effects by its ability to bind to many other effector proteins. One of those effectors is a protein called, shown here is MDA, also called diaphanous in humans. And diaphanous is known for its ability to stabilize microtubules and to regulate the polymerization of actin. So we studied this protein, and functionally this protein actually is inactive in the cell, and it's inactive by its ability of the DNA, this autoinhibitory domain called DAD. It's bound to this GBD, or GTPase binding domain, and only in response to cell signaling, in response to the presence of activated Rho, do you get the release of the DAD from the GBD, and Rho can bind and allow for a change in the cell's cytoskeleton. So what Deng-Hui did actually is to, I obtained wild type and mutant diaphanous that was fused to GFP and transfected the H295R adrenal cells with these. And as you can see, this allowed us to see which cells were transfected in green and the cells that were not transfected. And shown here is actually the actual mutant, the constitutively active mutant of diaphanous. And what she found is that the cells that were untransfected exhibited that mitochondrial movement, but the transfected cells had the mitochondria that were changed. They changed the morphology, which had been shown before by another researcher, but they were unable to exhibit that movement. And these actual cells had altered levels of cortisol that were produced. So Deng-Hui went on to do a lot of mass spec, purify lots and lots of diaphanous from the H295R cells using an antibody to the diaphanous, ran several gels, and shown here just a Kamasi staining of one. She cut out all of these bands, digested them with trypsin, plated everything on a MALDI plate, and did mass spec to identify proteins that were bound to diaphanous. And of course, shown here, the top hit is diaphanous with other proteins that are known to be involved in the cytoskeleton, like the tubulin, actin, thionin, the microtubule-associated protein. Vimentin we thought was interesting because that recently has been shown if you knock that out in the mouse, they have insufficient cortisol, or corticosteroid, I should say, production, and the female mice are actually have impaired fertility because of a decrease in hormone production. So we're currently, Deng-Hui and a couple of undergrads in the lab are characterizing the function of several of these proteins by either overexpressing them or knocking them out and seeing how they contribute to the ability of the adrenal cell to make hormone. One thing that mass spec can allow you to do, not only to identify what proteins are there, but to identify if a protein is modified. So one thing that Deng-Hui found is that the diaphanous is phosphorylated. So we actually generated an antibody to one of those sites, and as you can see, that diaphanous is actually phosphorylated if you treat cells with dibutyrocyclic AMP and do Western blotting using a phosphospecific antibody that we made. You can see an increase in phosphorylation of diaphanous that can be inhibited when we inhibit PKA activity, suggesting that this is a signaling regulated process. So we are still working on this. For example, I have an undergrad in the lab that's looking at ORP2, which has been shown to bind different lipids, steroids, sterols, and she's trying to answer the question of whether ORP2 actually is the protein that binds to the 11-deoxycortisol and carries it back to mitochondria, or maybe it's playing some other function. Then I have a graduate student who is actually looking to see if these two membranes actually fuse to allow the 11-deoxycortisol just to migrate back to mitochondria in response to activation of the signaling pathway. So we're still trying to characterize the function of all these proteins, but we know that the affinis-1 and its phosphorylation, that phosphorylation actually serves as a timer to mediate its degradation. So we're studying the function of all of these other proteins that she's identified. So I have to acknowledge the people that did this because I definitely didn't do anything. So in the first part, I talked about the studies with ASH1. Those were carried out by an extremely talented graduate student who just got her PhD and left me. Natasha Luque from Brazil, actually. And she's now a postdoc at the Scripps Research Institute in La Jolla. And she did these studies in collaboration with Kai, a postdoc in the lab who's working on the role of phospholipid signaling in the nucleus. And then Donghui Li, a very great postdoc in the lab, she did all of that real-time video imaging. And at the time, you pay for everything that you do. So real-time video imaging, it's a microscope, a really high-end microscope that we didn't have in the lab itself, because it's like $1 million scope. And Donghui, to save me money, would do these experiments from 10 p.m. to 4 a.m. because it was cheaper if you signed up to use the microscope at night. So she made all those movies for months and months and months. That's dedication, that's science, that's hardcore. Are you committed? So she would do all those movies at night to determine the role of these signaling pathways and that mitochondrial movement. And then Tamara, she's an undergrad, actually she's at Cal State Dominguez Hills, but she spends the summers with us. And she's been looking at members of that complex and is actually writing a paper right now on her studies with the ORP2. And the last people I have to acknowledge are Shurma Nuramaya in Japan. He gave us that construct, the GFP tag, wild type and mutant diaphanous constructs. Al Merrill at Georgia Tech, he was my former colleague before I left there. He is a sphingolipid guru who does mass spec on sphingolipids. So he did all of our sphingolipid mass spec. And a former student of mine, a graduate student who's now a postdoc at Emory, Eric Dahmer, who did the mass spec to identify the diaphanous-bound proteins. And of course the NIH and NSF for funding. So the last thing I have to say, which is important, I forgot to say, for people, since this is a group of students primarily, things that I figured I'd spend not more than a minute, hopefully, telling you things that I've learned along the way. So I know you all are trying to figure out, where you're going to be, what you're going to do, looking at everybody else who looks like they have everything all planned out and ready to go to medical school and be this great doctor or this great whatever. I've learned, never, rules from Marion, never be afraid to fail. That's one thing that it happens in the lab. We do many, we've blown up things in the lab. We've done all kinds of things that happen in the lab. Never be afraid to fail, whether it's an experiment or what can at the time seem like a really bad choice, like a bad choice in a lab that you spend time in, or a bad choice in a major. Never be afraid to fail, and never be afraid to change that. So if it's not working for you, change it. So if you decide that, hey, I decided when I got my degree, when I was a sophomore, I did my research, I was into organic chemistry. I just loved it, loved it. Took all the organic chemistry I could, and then did a research experience in a lab in organic chemistry, and realized that I could not spend the rest of my life doing this. So I changed that. And instead of going to graduate school and getting a PhD in organic chemistry, that was my plan, I did a research experiment my last semester before graduating in a more biomedically related lab that worked in the kidney, actually. And I knew that that was not what I wanted to do in graduate school, but I knew I wanted to do something more biologically related. So I got my degree. Well, before I got my degree, when you look through the brochures of whose lab you want to rotate in, I said, wow, nicotine addiction, primates. Wow, this is so cool. And at Emory, they have the big Yerkes Primate Center. I can work with animals, and this is going to be great. And I rotated, and I was like, You know, if I spend one more day watching a chimpanzee hit a button to get more cocaine, this is, you know, I'm going to shoot myself. I mean, this is not something I could do. But, I mean, some other people, most people in the lab loved it, and they still do that type of research. So you have to do something that, find your niche, and never be afraid to change it. Because I thought in high school I was going to be a marine biologist, because I'm from the Virgin Islands. I mean, who doesn't want to spend their time in sand? Me. Actually, I love looking at the sand and boating, but not researching and the smell of fish and seawater, and it's like, no, I'm too girly for that. So whatever you do, whatever you decide, never be afraid to change that path, because there's so many opportunities out there that you really will be amazed at what you can accomplish when you're happy doing what you want to do. So if you have any questions, I definitely will take them.